Hi, this is Tia. My apologies for not being able to present this in class, but here is my presentation. So to control the insect vector of human malaria, it requires development of an endonuclease-based gene drive system that interferes with the mosquito ability to transmit the disease, which could be achieved by either blocking parasite development in these mosquitoes or by reducing the reproductive capability of the insect vector. So on my paper, I chose to focus on reducing the reproductive capability of the insect vector. So the title of my paper is CRISPR-Cas9 gene drive engineered on female Anopheles gambiae mosquitoes' reproductive system, and the goal was to study how they eradicate mosquitoes carrying parasites specifically using the CRISPR-Cas9 gene drive on the reproductive system. My outline have an introduction which will explain why eradication of mosquito is important. It will explain what's CRISPR, and it will explain how CRISPR helps eradicate mosquito. This section will be very brief because I'll also go into detail again with these topics. In my three main points, it will focus on CRISPR-Cas9 targeting female mosquito reproduction. My first main point will focus on explaining the three genes that influence reproduction and why these three genes were ideal. My second main point will explain how CRISPR edit these genes and how they knew where to insert these genes. And my third point will explain the consequences of elimination using CRISPR in the ecosystem and how it affects the species around us. And then it will have a conclusion, which will summarize the data and the results from this section, from these sections. So for my first data, it will have my first data have three sections or three graphs, but I will only be focusing on A and B. So in A, to explain what's going on here, the ATTP is a guide plasmid, the 3 times P3 is a promoter sequence, GFP is a green fluorescent protein, and of course, the plasmid contain the DNA, the chromosomes that we are influencing. So A is an example of what they will be doing, but before they inserted Cas9 into the gene, they wanted to test to see if the three genes that are involved in reproduction were ideal or were good genes for homing. Would they be able to insert a gene into it, and would that gene be able to replicate into all the other genes that are also in the mosquito's body? So what they did was they used the green fluorescent protein. They caused a double-stranded break homology, and they inserted into the target sequence to see if the gene will replicate into it. And what they did was they used CRISPR-Cas9 nuclease to selectively disrupt the coding sequence of these candidate genes and analyze reproductive phenotypes to validate the suitability of these genes as homing target. And what they noticed that it had high levels of homing, which is it was able to replicate again in other genes. So it had high levels of homing in all three genes, because all three genes show sterility, and that's what they're looking for to that's what they're trying to do in these mosquitoes. So if we go to C, what we see here is the plus plus is a wild type mosquito, and the larva progeny, them producing eggs with no gene change. So this is just mosquitoes with no gene influence. In the sidebar, you can see the three different genes at the side, and each three different genes have a heterozygous and a homozygous gene. So what you can see is every single in each gene that had the heterozygous, they produces egg because the mosquito are still able to produce and or lay eggs with one copy of the edited gene. But when you see that they had the homologous two copies of the edited genes, they were unable to lay eggs, which showed that they needed the Cas9 system in the sequence, because the Cas9 sequence in the system insert that specific gene in that sequence into all genes, so all genes would have this the same sequence, and it would be homologous. So in this A, as you can see, is where they insert Cas9. This is a red fluorescent protein. And as you can see, when they insert the Cas9, it was able to replicate in other genes and homoanalous and became homologous pair. And as you can see, B also showed that it was able to take the one copy that was like the Cas9 one copy and was able to replicate in other genes and make it homologous in two copies. So it wasn't heterozygous anymore, it was homozygous, and that's what a Cas9 does. It replicates in target genes and go throughout the body. So what they saw was using the Cas9, it was able to take a gene with one copy and replicate it into more genes. And as generation, we see that the one copy of gene, the mosquitoes now had two copies of that same gene in their gene. They had that one gene in all of their genes because it was able to replicate and cause a double-stranded break and insert into all the target genes that they selected. And in mosquitoes that didn't have that was wild type, it was able to um, pretty much overpopulate that gene arc. And what they saw was, because of the Cas9, the population of mosquitoes that they were able to edit showed a 98% um, homologous gene, all of them in the third generation. So by the third generation, all of these mosquitoes either had that didn't have a copy had a gene, now a copy of at least one gene. So in the next generation of this, it will have two copies, and in generation that had one copy had two copies because of the Cas9, because it was able to replicate and transfer into all of the target genes that it target. So hopefully I explained this and explain the purpose of Cas9 and how it is able to um, replicate in target genes and make them homologous. And I hope I was able to explain that without homologous pair of this target gene, the mosquitoes are still able to produce and lay eggs. So they need the two copies of this homology directed repair green fluorescent protein gene to be able to become sterile. This is my reference.